So Carlos will be talking here about supplementing with adult coho to enable natural mate selection in the Russian River. Would you all please welcome again Carlos Garza.
Thanks. Actually, much louder. Strictly speaking, what Greg just said is not entirely accurate. The adult release supplementation that I'm going to be talking about today involves fish that were reared in the Russian River but not released there, although those adult release, or the adult release strategy is now in use in the Russian River as well, but those are not results that I'm going to be presenting today. Let me try this. So, here, let me give you that. Cheers. So, we've been talking most of this session about coho salmon from either here in the Klamath Basin or even further north, and spent relatively little time until Ben White's excellent overview of the Warm Springs Hatchery Coho Salmon program, have not really talked much at all about the southernmost of the coho salmon ESUs, the Central California Coast Coho Salmon ESU, which extends from just south of the Lost Coast down to Santa Cruz, California, where we have the southernmost persistent populations of coho salmon in the native range. And as probably all of you know, the demographic declines that have been observed here in the sunk ESU have been even more dramatic in the Central California Coast ESU, and they started earlier, and the status of populations in Central California is actually a lot more dire than they are overall in the sunk ESU. And you can see here on the map the number of streams with recent records of coho salmon in them, and these are, one observation in an entire tributary would lead to a line here, versus what is present, or what is recently present in the CCC ESU. Now, I'm not going to belabor the background material associated with what's going on in the CCC with respect to the use of hatcheries as a conservation tool. You've heard a bit about that already. You haven't really heard much about the Scott Creek program that we initiated just a year after the Warm Springs program. It's a lot smaller scale. It's frankly not been as successful as the Russian River program. So I'm not going to talk much about it, but and I'm going to focus on what we've been doing in Marin and Sonoma counties associated with the Warm Springs Hatchery Russian River program. And I've outlined the basin here, and I've also indicated the two focal streams that we're going to be talking about, two of the focal streams we're going to be talking about today, as well as Santa Cruz down there at the southern end of the range. So this is the study area that I'm going to be discussing today. It's nested right in the middle of the CCC ESU, and this is the lower portion, most of the coho habitat indicated, in the lower portion of the Russian River here, with Warm Springs Hatchery on Dry Creek indicated, as well as Salmon Creek, which is just to the south of the Russian River Basin, a small coastal basin that flows directly into the Pacific Ocean right north of Bodega Bay, Bodega Head, as well as streams in that are tributary to Tamales Bay, which separates Point Reyes National Seashore from the rest of Marin County in Central California. Walker Creek, where coho salmon have been absent for several decades, as well as Lagunitas and Lima Creek, which, as was mentioned earlier, are sort of synonymous. They meet kind of in the estuary right here. This is all sort of estuary, and are two tributaries of the same basin, and we frequently use them interchangeably. Sorry about that. And we're going to talk about this adult release strategy and the observations and the experience that we've had with this strategy in, again, these two basins here, Walker Creek, where they've been absent for decades, and Salmon Creek, where ironically salmon have also been absent for decades, or at least were until the beginning of this program. So I'm just going to tell you a little bit about the genesis of this project, and it was really almost kind of by accident. When we started the Russian River program in the early part of in like 2001, a decision was made to collect a number of fish from Lagunitas-Salima Creek, bring them into the hatchery as potential broodstock for the supplementation effort in the Russian River. There were some preliminary genetic analyses and some other considerations that led us to believe that it was not prudent at that time to be introducing fish or genes from the Lagunitas-Salima Creek into the Russian River population. So we had all these fish on our hands that we were not going to use. They had been raised their entire lives in fresh water, taken as juveniles from Lagunitas-Salima Creek. And the Lagunitas watershed people did not want them back. They were concerned that there were going to be disease issues and maybe some effects of having lived their entire lives in the hatchery. So we had to find something to do with them, and there was consideration of just dumping them into the ocean or whacking them on the head or whatever. And about that time, a friend of mine had taken over as the manager of a Marin County Department of Education outdoor education facility on Walker Creek Ranch. And they had been involving the community in habitat restoration and were restoring habitat, but didn't really have any fish. And so they quickly agreed to allow us to release these fish into Walker Creek, the Walker Creek Ranch. And nobody really knew what was going to happen because we hadn't done this before, and these were fish that had lived their entire lives in fresh water. And lo and behold, so we released them starting in 2003. At that, I think the first year there were approximately 80 fish released, and the next year when we came back, or our partners came back and monitored, they found juvenile coho salmon. Only 8 of them, 7 in one trib and 1 in another. And they took small fin clips non-lethally, sent them to us, and we were able to confirm that those 7 and from the one tributary were the offspring of the hatchery fish, the hatchery reared fish that were released there using parentage analysis. However, there was one fish there that could not be accounted for, one of the juveniles could not be accounted for by the parents that had been released, and we still don't know what the origin of that fish is, and we've speculated wildly about whether or not the presence of adult fish in Walker Creek may have led to some olfactory attraction of other natural fish and instigated spawning there by fish that otherwise may have not entered there, since we hadn't seen any fish there in spite of intensive efforts to find them for decades. On the basis of that work, there were adult releases that ensued in a number of years, and we were able to determine either directly or indirectly that those fish were largely a result, if not entirely a result, of the adult release program or projects that we had done in Walker Creek. And Walker Creek is a little bit special because it empties, for a number of reasons, it empties into Tamales Bay, right at the mouth of Tamales Bay. So all the fish that are going to Lagunitas-Salima have to swim right through the outflow. And so it's a little bit different in the sense that there may be some effects, some attraction effects there that would be not necessarily be occurring in other places. But the other thing that's interesting about it is that it's actually a Superfund site because of an abandoned mercury mine that's been leaking mercury into the watershed for a number of decades. So on the basis of this project, we decided to expand it to Salmon Creek, where again, there hadn't been any coho salmon seen since approximately 1980. And because of the intermediary location of Salmon Creek, more or less equidistant between Lagunitas and the Russian River, we hypothesized that historically that basin would have received migrants from both the Lagunitas, Tamales Bay complex as well as the Russian River. And at the same time, we were dealing with some quite serious inbreeding issues in the Russian River program and trying to figure out whether we might be able to use outbreeding, outcrossing has been referred to it, as a way to ameliorate the inbreeding problems. And so we tried something that was really very experimental in nature and actually turned out to be quite informative. And it was experimental and interesting enough that it actually made the front page of the San Francisco Chronicle. And what we did is to release fish from those two locations in roughly equal numbers in Salmon Creek to see what would happen, see whether or not there were natural mate choice patterns could inform what we should or should not be doing in the hatchery. And I'll circle back around to that. So ironically, the next year, it was a bad water year in Salmon Creek, and the Department of Fish and Game had to go in and rescue a bunch of juvenile fish from a couple of the tributaries where they had been planted. And we got tissue samples from those, Fay and Finley Creek, and we genotyped them with the same genetic markers that I talked about so extensively yesterday, and analyzed the data in a variety of manners, and got some answers. And what we found is that in both of the streams, the predominant class of juvenile fish were fish that were hybrids between the Russian River and Lagunitas and Lima releases. Very few of the fish actually came from Russian by Russian crosses, and a moderate number, but still less than hybrid, of Lagunitas by Lagunitas fish. And we can translate that into matings, and keep in mind these are minimum numbers and just for a small fraction of the fish that were released because they weren't all released in Fay and Finley Creek. And we were able to confirm differential numbers of matings as well. So a couple of things I want to point out here. One is that we don't have enough statistical power here to determine whether or not there is a predilection for outcrossing in in this natural setting, hybridizing, if you will. But there's certainly a trend in that direction, and we also see a substantially larger number of juveniles per family group in this hybrid class than we do certainly in the Russian River, and it's even a bit higher than it is here for Lagunitas Lima. So that in the absence of more analyses of this type, we can't say anything with statistical certainty, but clearly the fish are not avoiding outcrossing, and they may in fact be preferring it. Well, so I mentioned earlier that we had this big inbreeding problem in the Russian River broodstock, and it's a little, so the way that we had been doing mating in the Russian River program was not set up as an experiment to evaluate the effects of inbreeding on survival. In fact, we didn't feel that we had the luxury to have any highly inbred class that we could use as a control to evaluate survival. And remember, we were almost entirely eliminating highly inbred matings in that stock, in that program, through use of the broodstock, the spawning matrix. Even so, when we did generalized linear modeling, and this is work that Luis Conrad, who had Ben's position, before he did really spearheaded. When we did modeling of the primary determinants of juvenile survival in the hatchery, what jumped out at us as the significant factor was the relatedness coefficient between parents. So even when we're truncating the distribution of inbreeding coefficients, if you will, and only allowing fish that are maybe related but not at a full or half sibling level, we still see that residual inbreeding is still causing a significant effect on survival in the stock. So an inbreeding problem for sure. So as I mentioned, we initiated about that same time outcrossing, and Ben described this quite a bit, with fish from the Lagunitas Lima stream population at a relatively low level in the Russian River broodstock program, and were able to determine, and these are just some representative plots, and we're able to determine, in fact, that the outcrossed fish survive. I didn't show you the data on this, but it's so striking. You can actually pick out the hybrid matings when you go look in the incubation trays. You know, one of those things that you don't really need statistics to know is a different in hatchery survival. But what these three bars represent are, number one, these are Russian River by Russian River crosses, and unfortunately they're in different colors in the different graphs. Sorry about that. Always the first one is the Russian River, the inbred mating, if you will, and then these are the two reciprocal matings, Russian River males by Alima Creek females and vice versa. And in fact, we were able to demonstrate a significant increase in both overwintering survival and oversummering survival post-release in the Russian River prior to outmigration of these outcross salmon. So outcross salmon survive better at all juvenile life stages. And this is, I think, fairly compelling evidence that, that these fish know how to avoid inbreeding. And when we bring them into the hatchery and we don't give them that opportunity, we are causing problems that can be alleviated through the use of a variety of methods, including broodstock management and the careful incorporation of fish from other closely related stocks. So in summary, endangered coho salmon raised, and this is endangered in the legal ESA sense, raised entirely in freshwater, reproduced naturally in streams when released at maturity. We think this is true for fish that have returned from sea, hatchery origin fish, but we don't actually have the monitoring data yet to determine that. We know that fish of that type are put in streams and that reds appear. We just haven't closed that loop yet. In Salmon Creek, the majority of the surviving offspring from these adult releases are Russian River by Alima Creek hybrids and the result of spontaneous outbreeding. The salmon that we release there do not avoid hybridizing with fish from similar populations, and they may actually prefer it. More data necessary to make that a statistically supported statement. And we've been able to determine that this outbreeding, hybridization, if you will, has a direct positive effect on survival of the juveniles produced, not just in the hatchery, but also in the natural areas post-release. And again, we need to close the loop and look at the marine survival and the adult returns of this as that becomes possible, and we'll be doing that in the next few years. And again, we were able to confirm that and this is sort of a little backwards, but both in Walker Creek and in Salmon Creek, the juveniles that we've observed are the offspring of the adults released in the previous winter. So we know that these fish are not just coincidentally appearing, being produced in the streams where we release these fish. We've done a couple of direct analyses. And as a consequence, these adult release projects are now occurring in a number of different locations, including in the Russian River itself and the two streams in Marin County that I've discussed, and at a number of streams in Santa Cruz County where with the smaller program that I mentioned at the beginning of the talk. And again, we just started the adult release procedure in Santa Cruz County this year, and everywhere we put them, we've seen reds. And those reds are most likely, but not yet demonstrated, to be from these adult releases. They're not a whole lot of natural origin fish, or any in some of those streams. So this is a very powerful, low-cost, low-tech, low-risk, and potentially very important strategy for supplementing natural populations in a way that allows mate choice to operate, the non-fit fish to not leave offspring, and allow that, what I like to call the reconditioning of genes of hatchery reared fish to start one generation earlier than any of the techniques that require spawning of fish in hatcheries and then releasing juveniles. And this is, I kept saying we, and that we is really a very big we. A lot of people were involved in this. I actually never took any of these fish myself and put them in the streams, but a number of people, including Ben White, have. And so everyone, I can't even acknowledge everyone that's been involved here, but a number of them are. And with that, I'll thank you for your attention.
Thanks, Carlos. We do have time for questions. I'll let everybody know.
Hi, Carlos. Wakefield Fish and Game. One quick question for you. I was wondering if you had any hypotheses on what the mechanism is for this inbreeding avoidance.
Well, it's actually been demonstrated in every animal where it's been looked for. The mechanisms are probably olfactory in nature. We know that in some fishes, for example, that they have the ability to smell, if you will, the immune system genotypes. The immune system genes in vertebrates are typically the most variable in the entire genome, and over the last 20 years we've come to recognize that variability serves more than one function. It's not just a way for increasing the ability of an organism to respond to different parasites and pathogens, but it also turns out to be important in individual recognition. And for example, it was shown almost 20 years ago in a series of papers in Science and Nature that mice use the smell of the immune system genotypes to both avoid mating with closely related, I was going to say fish, other mice, and also to identify close relatives to create communal nests. So, this has been shown to be the case, although for some people not conclusively in fishes. So we believe it's an olfactory mechanism that's probably being driven by immune system or other highly variable genes. So,
Rick, you have a question, and then Keith.
From a practical standpoint on the adult release thing, I see the ability for mate selection being an advantage, but given that a lot of these programs aren't going to have a lot of fish to do that with, maybe 10, 20 pairs, when you've got a limited number of potential mates to choose from, if all you got's ugly ones or unfit ones, you're still going to choose somebody. I mean, that's the way it works.
well, that's right, yeah.
And so there in terms of the number of adults that are let out there, there's still an artificial selection involved in those adults. And so I'm wondering on the advantages of having to really achieve the full effect of mate selection, having to have so many adults in there so that there really is a lot of choice for mates, versus artificially doing it in an eyed egg or some other type of thing where you've divided up the milt and the eggs and divided up and achieved as much diversity as you possibly can. Can you, did I make myself clear?
I kind of, yeah, but I think, it's not, we as human beings are not as good as fish are at determining what the important types of diversity are when one chooses a mate. And even if we do what we're doing now with the iron gate broodstock, the Coho broodstock, using genetic broodstock management, we could very well be missing things that are not apparent just by looking at this marker data, genetic marker data, but that are really important to fish when they're choosing mates, and they could have direct selective consequences. It's true that in my view, really sort of the only downside of the adult release strategy, and it's some people would actually consider it an upside, is that productivity of those adults relative to being spawned in a hatchery is going to be a lot lower. I didn't talk about this because we don't really have all the data to nail it down, but in general we know that most of these, well, no, not most, that some, and in some cases maybe most of these fish are not actually reproducing. Some are. And one could make an argument that that's a negative thing, and you're losing productivity that you might be able to retain if you're spawning these fish in the hatchery. But the flip side of that is that maybe those fish are ones that are not fit enough to produce in the wild anyway, and those are really not fish we want as part of the natural population. Those are two arguments for and against it. But the other thing that I would say is that I'm going to go from sort of reporting on this to a personal observation about what we're discussing here. That wouldn't apply in the situation that we've discussed here in the Klamath Basin, where you've got hundreds of fish that are, well, until 2 years ago, were coming into the hatchery and not being spawned there. The natural origins were being put back. The hatchery origins were being whacked on the head. And that productivity was zero, well, at least for the hatchery origin fish. And so it's, and the one last thing I want to say about that is, and this is what we've been doing in the Russian River, so we work with Ben before the releases to help him choose from amongst the very many individuals that he has the most diverse set of fish that can be literally stratifying the sample to go in for the adult release by families to make sure that we're getting as diverse a set of fish into the stream for natural spawning as possible.
So your opinion then, to summarize that, your opinion, it's better to have limited volitional mate selection than no volitional mate selection at all, as opposed to some of the other things where it's just completely humans parceling up the eggs and milk?
If your intention is to recreate or supplement a natural population, yes. That's my opinion.
Keith, you're next.
Carlos, if you didn't have me confused enough yesterday, you've done it today.
Sorry.
Well, yesterday we were talking about divergence of the hatchery stock compared to the natural origin spawners, and we're trying to minimize that. Here it appears that you are doing the opposite of that, and the hybridization has better survival rates. What would be our goal in the supplementation here at the Klamath Basin?
Oh, those are kind of two separate questions. What's going on in the Russian River was the outcrossing was a response to an observed and easily observable problem that was leading to decreases in survival. And remember, this is intended to be a natural population supplementation program, the Russian River program. And we knew that inbreeding was a problem. So we kind of felt like we didn't have much choice. We needed to try something because inbreeding is deterministic and it leads to extinction. It's not, there's not much mystery about that. So, and so we were cautious, and it took us, 5-6 years get to consensus about whether or not we would allow some genes from Lagunitas lima to be integrated into the Russian River population. And then as soon as we did, and everybody, I mean, literally immediately could see this jump in the early life stage survival and the hatchery. I mean, deformities just disappeared, and they were, well, not entirely, but, and survival just was hugely different. And so that sentiment changed pretty quickly. And the goal again is to increase the number of founders, to get those genes mixed up into the population, allow natural selection and adaptation in the natural environment to drive productivity and allow the reemergence, if you will, of the ability to produce in these natural areas.
Does that mean that would be introducing out of basin genetics at the hatchery?
Well, you know, here in the Upper Klamath Basin, there's still a bit of material left, and the situation is not as dire, anywhere near as close, if you consider the entire Upper Klamath as a whole. In the Shasta itself, it's pretty close to as dire. And in fact, it's, I would say, pretty similar if you consider the Shasta as just an isolated sub-basin within this larger metapopulation, which, as I think was mentioned yesterday by Sean Narum, but we as geneticists typically do not. We do not think of these basins as being completely isolated populations that are, you know, sort of, the goal should not be to retain them as entirely pure and untouched by genes and fish from other nearby tributaries. That's not consistent with what we know about salmon biology. So,
Thank you.
See, when Ben was talking, he said that he was talking about how when the adults have some problems with their air bladder and their physique when they're being released. And I was thinking about the fish that were released from Iron Gate, the extra ones, and I forget if they were marked fish or unmarked fish. Were they unmarked or marked? Oh, okay. If they were actually trucked to the where we wanted them to spawn, do you think that they would have spawned there, or perhaps if, since they're held in captivity for a while, they lose some of their fitness as far as their muscle tone, and so you release them and then maybe they're too tired or too worn out to swim up the river, and perhaps they spawn in the canyon because they just don't have the energy to go any farther. And so I was thinking about that, and then about the releases of your fish. If you release them, you release the large fish, the adult fish where you wanted them to spawn. If you had released them downstream, would they have swam, completed their cycle and swam upstream, or would they have been too worn out from living in the hatchery for too long as adults?
well, yeah, I'm not a physiologist, so I'm gonna not actually answer the question about fatigue in fish and whether they have the energy to continue to migrate, because I don't really know the answer. But keep in mind that what we've been doing here is taking fish that are on the verge of spawning. They're reproductively mature. They're either already starting to express milt or just about to, and putting them directly in the habitat that we want to see them spawn in. So it's really, we're not, the intention is to have the fish that are released stay right there and spawn and not move all around. Now, with respect to what's happening with the fish that are released from Iron Gate Hatchery that are put back down in the pipe when they're not needed for spawning, well, we know at least some of those have gone up into the Shasta, and so we don't really know if they've spawned or not. That's something that can be evaluated with with careful monitoring. But it's important to point out, and this is relevant to something that I think Dave Webb said yesterday, that the expectation for the numbers of fish that might swim into the really high-quality spawning habitat in the Upper Shasta and the Big Springs complex, for example, is the numbers that we would expect to do that, even under sort of the best-case scenario, are just not enough to get the critical mass of fish that would be necessary to overcome these demographic issues that are really, I think, probably the biggest problem that faced by the Shasta Coho population right now. So.
Let's thank Carlos, and he will be here for the panel, so more questions then if we, so let's thank Carlos.